Which of the following is not a second messenger? A. CMP, B. PKA, C. Calcium, D. Diacylglycerol, and E. IP3. Think about it a little bit, and let's look through them one by one. So, starting with CAMP. Is it a second messenger? Well, obviously it is, right? It's a classic second messenger. And may I ask, what enzyme is it produced by? Yes, it is produced by the enzyme adenylate cyclase. And what does it do? Well, it travels through the cytoplasm to physically bind to and activate something, okay? It travels through the cytoplasm, but the question is, what does it activate? Think about that. If you don't remember what does it activate, you can look at the other answer choices, and it actually activates PKA, which is the second answer choice. So it activates PKA. So we know it's a second messenger, and we can eliminate that. Okay. And now B says PKA. Is PKA a second messenger? Well, let's see. So we know PKA, protein kinase A, it is a kinase, right? So it's actually an enzyme. So it is a large multi-subunit enzyme. So it is an enzyme. So that's first thing first. It's a kinase. A kinase protein. So because it's a kinase protein, it's definitely not a secondary messenger. So it's not a second messenger. So we know this one is the answer. So what exactly are second messengers, right? Second messengers, they're small. Right? Second messengers, let's put an arrow here. They're supposed to be small. And they're non-protein, right? The non-protein molecules or ion. That rapidly diffuse throughout the cytoplasm to pass some signal along. And PKA is actually the downstream target that gets activated when a second messenger binds to it. So when a second messenger binds, PKA, the enzyme kinase protein, gets activated. And since it's a kinase, guess what it does? It phosphorylates other proteins, right? So it's a kinase protein, so it's going to phosphorylate other things. Okay, so that is PKA. And then, so that's our answer. Now we look at option number C, which says calcium ions. Well, extracellular signals trigger it. So it's an ion, so that's actually a hint that it could be a secondary messenger. And why it is, extracellular signals trigger the release of these ions from the endoplasmic reticulum into the cytoplasm. So released from endoplasmic reticulum into the cytoplasm. And when they're in the cytoplasm, they bind to target proteins like commodulin to alter cell behavior. All right, so now we can look at option number D. So we can eliminate number C. Number D says diacylglycerol. Sometimes also abbreviated as DAG. So DAG is a lipid-based second messenger, right? So we know it's something that's after it's being cleaved by phospholipase C, right? It goes on to activate something, if we remember this pathway. And because it is a lipid-based second messenger, so lipid-based second messenger, it remains inside the cell membrane after being cleaved by phospholipase C, which is also known as PLC. So remain in cell membrane after cleavage by PLC. And what it activates, do you remember what it activates? Well, it activates protein kinase C. And also known as PKC, okay? All right, so finally we have IP3. This one is also called inositol triphosphate. And it's a water-soluble second messenger that's cleaved from membrane lipids alongside DAG. So unlike DAG, it is water-soluble, cleaved from membrane lipids, specifically PIP2, right? So it's cleaved from membrane lipids alongside DAG. And what it does, well, it relates back to option number C. It diffuses to the endoplasmic reticulum and opens calcium channels. Plasmic reticulum and opens calcium channel. All right. So that's it for this question. So the answer is that pKa, it is not a second messenger. Thanks for watching.